Good morning, everyone. Thank you for the invitation and opportunity to present today. The topic assigned to me is high-resolution brain function, imaging small subcortical nuclei and cortical layers. I'm sure you'll all agree with me that the brain is a fascinating human organ, which really makes us human. It comprises of more than 100 billion neurons organized in layers and or as discrete, large, and small nuclei within 1300 ml approximately of the parenchymal volume of the adult human brain. And of course, there are other cells in the brain, including an almost equal number of glial cells that support neuronal function as well. Diseases of the brain are a major source of disability and mortality burden worldwide, leading to thousands of disability-adjusted life years lost per 100,000 population. These include time lost due to diseases like stroke, dementias, Parkinson's disease, multiple sclerosis, epilepsy, traumatic brain injury, headaches, etc. A comparable number of disability-adjusted life years are lost due to neuropsychiatric illnesses, largely including depressive disorders, anxiety disorders, conditions like schizophrenia, substance abuse, etc. And these conditions are also widespread around the world. Unfortunately, the burden of many of these diseases is increasing over time, depicted here for neurodegenerative diseases. So there's clearly a need to better understand these conditions beyond our current state of knowledge, where we recognize the gross pathological changes in many of these conditions, such as the dementias, Parkinson's disease, multiple systems atrophy, and Huntington's disease, etc., and also recognize many of their microscopic features, which have been targeted in some cases for molecular imaging. These gross pathological changes have been harnessed by conventional imaging techniques to help with identifying and tracking some of these disorders. But there is clearly a need to go beyond these large structures and study the small structures in the brain, such as the small subcortical nuclei. The volumetric resolution of current molecular imaging techniques, like PET scanning, for example, is approximately 100 microliters. So I would like to suggest that structures that are less than 200 microliters in their volume may be considered as small nuclei. And there are several such small nuclei that are highly relevant for understanding not only normal brain function, but also various brain diseases. Some examples include the bed nucleus of stria terminalis with a volume of 134 microliters, approximately, parabrachial pigmented nucleus less than 100 microliters, subthalamic nucleus 135 microliters, ventral tegmental area, mammillary nucleus, and habeneural nucleus, and many others, which have very low volumes. These small nuclei are particularly tightly packed in this high-value real estate area in the brainstem, in the midbrain, pons, and medulla. Many of these nuclei have specific molecular signatures that have a role in pathogenesis of many of these diseases and need to be specifically studied in a discrete fashion. Similarly, as you know, the human brain cortex, particularly cerebral cortex, is organized in these various layers. The neocortex is organized in 6 layers. The cortical thickness can vary anywhere between 1 millimeter to 3 millimeters across the brain, depending on which region we are looking at. And the relative contribution of these layers to the overall cortical thickness also varies. The sensory cortex has more contribution from the layer 5, for example. The motor cortex has more contribution from the layer 5, for example, and the sensory cortex has more contribution from layers 1 to 3. And we'll go into a little bit more detail later. So we definitely need to study and image these small subcortical nuclei and cortical layers, because they are often involved early and in a specific fashion in many of these neurological and neuropsychiatric conditions. That can help pursue early diagnosis and differential diagnosis, which is a key issue in management of these patients. Similarly, what we consider as conventional neurologic or neuropsychiatric diseases are actually often a conglomerate of various conditions. So identifying these small structures and their involvement in these pathologies can lead to a better classification of these disorders, and then potentially an improved risk stratification and prognostication. which as a clinician, ultimately, I would hope would lead to improved treatment selection, planning, and monitoring. So my aim is to discuss a few examples today to illustrate some of these points. And the overall goal is to improve the understanding of the brain function and hopefully to contribute to reducing human suffering from these diseases. These small nuclei have a role not only in these neurological neuropsychiatric diseases, but also they're important to understand the normal brain function. For example, here, the pathways involved in sleep genesis are shown, which involve many of these small structures in the brainstem, hypothalamus, subcortical areas, and the cortex. One of the key regions I would like to focus on today is the locus ceruleus. It literally means the blue spot due to its appearance on neuropathology in freshly cut brain specimens. It is present in the base of the fourth ventricle, centered in the upper pons, extends into lower midbrain. It's the primary seat of noradrenergic neurons in the brain. Its dimensions are 2.5 by 2 by approximately 14 millimeters or so, leading to a volume of approximately 25 microliters. It's really a very small structure. However, it has widespread projections to the cerebral cortex and has role in wakefulness, arousal, attention, and has been shown to undergo early degeneration in conditions like Alzheimer's disease, Parkinson's disease, multiple sclerosis, and several other conditions. It may also have a role in immunomodulation in the brain, which has in turn potential huge implications for neurodegeneration and progression of the neurodegenerative pathology. I had an opportunity to image the norepinephrine transporter system in the human brain using the high-resolution research tomograph during my postdoctoral work at the Yale PET Center, where we delineated the distribution of the norepinephrine transporter in the human brain in the locus ceruleus and pontine raphe, where it leads to this wing-shaped appearance, the midbrain raphe, red nucleus, hypothalamus, and thalamus. We were able to quantify it and demonstrate here the binding potentials of the C11MRB tracer in these small regions. And also we attempted to quantify that in the thalamic subnuclei. Here you can see the comparison between the pathological and the anatomical position of the locus ceruleus and its appearance on the HRRT in this averaged image. And in my most recent work in collaboration with George Al-Fakri and others at the Gordon Center, we can also identify the locus ceruleus in a whole body scanner using the C11 Meaner or C11 MRB tracer. We've shown that the HRRT could be used to identify differences between healthy controls and patients who suffered from cocaine abuse in these small structures, including thalamic subnuclei, locus ceruleus, and the thalamus itself. And we concluded that these results were consistent with the results obtained on neuropathological studies in humans and animals. However, we did recognize that we are clearly underestimating the concentration of norepinephrine transporter in these structures due to their small size. And we emphasize the need for nonlinear image co-registration methods, which have since been developed, and also the need for multimodality imaging, including inclusion of MRI for ROI placement for locus ceruleus. And with the hope of the ultra-high resolution PET scanners becoming available, we are very optimistic that would lead to an improved quantification of these small structures and better delineation of their pathology. Locus ceruleus is also potentially crucial for understanding Alzheimer's disease. According to neuropathological literature, the locus ceruleus involvement may be the Alzheimer's disease ground zero. Recent work in Parkinson's disease has also shown involvement of the noradrenergic system in patients who had REM sleep behavioral disorders as compared to those who did not, and as compared to healthy controls using a similar tracer. Another small nucleus that I would like to emphasize today as an example to illustrate the importance of these small structures and importance to image them using molecular imaging is this pedunculo-pontine nucleus, which is present in upper pons. The rostrocaudal length is only 5 millimeters. It is rich in cholinergic neurons in its lower 2/3. It is often a target for deep brain stimulation to treat freezing gait in Parkinson's disease. However, the lead placement is based on anatomical landmarks, although aspirational attempts have been made to identify it directly using 7T MRI here. Molecular imaging of the peduncular pontine nucleus has obviously been limited because of the lack of high resolution imaging uh, machines. Here there is a vesicular acetylcholine transporter ligand that concentrates in that area because of the presence of cholinergic neurons, but using an HR+, but clearly an improvement in resolution will lead to improvement in the quantification of this structure. And the deep brain stimulation, for example, the benefits are highly variable, and improved delineation of this structure can lead to an improvement in patient outcomes. There is heterogeneity within the pedunculopontine nucleus itself along its rostrocaudal length and its horizontal diameter. And therefore, a higher resolution imaging of the nucleus will definitely aid in more precise targeting of this region. I would like to also emphasize that in addition to the gray matter regions, one should also focus on the brainstem white matter tracts, which are tightly interspersed with these gray matter areas in the brainstem. Staying in the posterior fossa, the cerebellum, along with the cortex and white matter, has these small, deep cerebellar nuclei, which are affected differentially in different conditions. For example, here in pure cerebellar cortical atrophy, there is involvement of the cerebral cortex, only late involvement of the deep cerebellar nucleus. On the other hand, in olivopontocerebellar atrophy, in a patient with MSA, there is involvement of the cortex and then a cerebellopetal involvement of the deep nuclei, as opposed to dentorubral atrophy, where there is first involvement of these deep nuclei, and that can help distinguish these conditions from each other, and therefore high resolution imaging would be very helpful. For a condition like multiple systems atrophy, precise identification of pathology in these small nuclei can be very helpful in tracking these diseases, tracking this disease over time. For example, olivopontocerebellar atrophy here in the case of MSA spares the so-called fleece of the dentate nucleus in the cerebellum. And there is selective involvement of different parts of the brainstem and the pontocerebellar peduncle, middle cerebellar peduncle in this condition. The thalamic subnuclei also play a key role in different diseases in a disease-specific manner. More so, these nuclei may even have their own layered structure, as shown here for the lateral geniculate nucleus. And the scale here is 1 millimeter, so one could think about potentially distinguishing the layers within the thalamic subnucleus here in the lateral geniculate nucleus. Here are the various thalamic subnuclei, which are connected with specific areas in the cerebral cortex. And in our recent work using glial markers, we saw that specific areas of the thalamus were linked with either clinical measures of disability or with global brain atrophy. The vesicular acetylcholine transporter imaging that I talked about just now also showed this focal uptake in the lateral geniculate nucleus using the HR+. And clearly there is value in identifying these nuclei using a high resolution molecular imaging technique. The bed nucleus of stria terminalis, or the so-called extended amygdala, is a very small structure, and this is our recent exploratory work regarding depression in MS patients. And we find high tracer uptake in this area, which is essentially an overlap between the fornix and the BNST. And therefore, this is an example where a higher resolution information would help us separate the fornix from the BNST and help us make more informed inferences about the involvement of these structures. Cerebral cortex, as we discussed, is organized into these layers, and I already mentioned the variability in the cortical thickness in different parts of the brain. Interestingly, the functional unit of the cerebral cortex is thought to be this vertical column, which depends on the extent of the dendrites of this large pyramidal cell. And it lies in the size range of 0.5 to 1 millimeters. So again, potentially better estimated with higher resolution techniques, and could be potentially highly useful in a condition like epilepsy, and also for a correlation with modalities like electroencephalography, which depend on this vertical alignment of the cortical function. In terms of diseases and the value of understanding cerebral cortical layers, Alzheimer's disease and frontotemporal dementia tend to start first in the outer layers of the cerebral cortex and then involve the entire cortex. So therefore, identifying pathology in the outer layers could help identifying these patients early, which is a key factor in potentially determining their response to curative therapies in the future. In terms of neurochemistry, different neurotransmitter receptors or transporters are expressed in different layers of the brain in a different fashion based on their location. For example, here, the noradrenergic alpha-2R receptor is localized more to the middle layers of the cortex in the occipital cortex, but is more diffusely present in the motor and sensory cortex. And understanding this variability and then applying it to individuals using a higher resolution techniques would be highly helpful. Multiple sclerosis is conventionally thought to be a white matter disease. However, there is clear gray matter involvement based on pathological literature. And subpial demyelination is considered to be a specific feature of MS pathology based on pathological specimens. So identification of such layered pathology in the cortex in MS can be of high value as well. So I hope I have presented a few examples of the value of this subcortical small nuclei imaging and imaging of the cerebral cortical layers. And obviously we can attempt to do that based on improved detectability by improving the intrinsic contrast or contrast with specific molecular imaging agents. But in addition to just improving the detectability, we also want to improve the quantification by improving the scanner resolution and reducing partial volume effects. I cannot overemphasize the need for multimodal imaging so that all of these techniques can complement each other, covering for the other techniques' weakness and contributing their own strengths. And also it would be important to provide or to develop improved anatomical delineation methods such as atlases to help identify these small structures on these imaging modalities. I thank my mentors, my collaborators, my team, and the funding agencies, and I thank you for your attention.